Hi, my name is Samantha Kreltszheim, and welcome to my presentation on chloride. Today I'm going to focus on the importance of chloride ions in the central nervous system and skeletal and smooth muscles. Gamma aminobutyric acid, also known as GABA, is involved in how rapidly neurons fire and is the main inhibitory neurotransmitter. GABA plays a major part in modulating or mediating most of the central nervous system functions. GABA displays a wide range of pharmacological effects such as hypnosis, muscle relaxation, amnesia, cognitive enhancement, and more. Therefore, manipulation of GABA communication is of benefit in the treatment of many psychiatric and neurological disorders. How does GABA affect neural excitability? Pain impulses are generated at different points between the site of tissue damage along the afferent sensory fibers and within the brain and spinal cord. The neurotransmitter system may either enhance or reduce the propagation of pain impulses given the widespread distribution of GABA neuron activation. The afferent peripheral nociceptors are located in the lateral portion of the dorsal root. They enter the spinal cord and synapse in the marginal zone. These regions include cell bodies of the secondary neurons that develop to higher levels. Substance P and glutamic acid are the two main excitatory neurotransmitters released at the synapse. Valium, also known as diazepam, is prescribed to alcoholics/problematic drinkers as a sedative during withdrawals, although is contraindicated with alcohol use. Both these substances have similar pharmacological properties such as amnesia, disinhibition, and impaired judgment by acting upon the GABA receptors. They both induce similar effects, especially at high doses. Chloride ions are vital for correct physiological function, including electrical signaling in muscles and regulation of ion homeostasis. They operate through opening/closing devices, which are different to those of calcium, potassium, and sodium channels. Chloride ion channels are accountable for chloride movement. Sensory neurons accumulate a large amount of intracellular chloride concentration. The opening of a chloride channel advances to an efflux of chloride, thereby generating increased excitability and membrane depolarization. As you can see in this diagram, due to damage or inflammation and through the gating of inositol triphosphate receptors in the endoplasmic reticulum, intracellular Ca2 levels are raised. This Ca2 rising leads to the opening of ANO1 or perhaps other CACCs, leading to an efflux of chloride, resulting in depolarization and increased action potential propagation to the spinal cord. Channelopathies are diseases that develop as a result of the weaknesses in ion channels triggered by either acquired or genetic factors. Mutations in genes encoding ion channels, which damage channel function, are the most common source of channelopathies. Ion channel weaknesses have been connected to a wide range of diseases, including epilepsy, blindness, deafness, diabetes, cardiac arrhythmia, cancer, and many more. A channelopathy that affects the conduction of chloride in the muscle is myotonia congenita. Because of delayed relaxation caused by sustained electrical activities in the muscle, this is present with attacks of extreme muscle stiffness. This is a result of loss of function mutations in a single gene, which encodes the skeletal muscle chloride channel to steady the resting membrane potential and influence the membrane repolarization following action potentials in skeletal muscle cells. Loss of CIC1 channels decreases the inward chloride current requirements to compensate for the depolarization induced by the potassium accumulation in the transverse tubules, resulting in spontaneous recurring firing of action potentials and a slower degree of repolarization. Thank you very much for watching, and I hope you enjoyed my presentation on chloride. Thank you.